Testing. So I guess we can try this one. Testing. All right, this one works. Hello. Hello. Can you hear me? Some of you can hear me. I've been working on the audio setup for the past 5 minutes, but I think we have something that works. Are you all here for computational neuroscience? Okay, good, me too. So yeah, I'm Michael Rule. I'm the unit director. We also have two, I want to say teaching assistants, but I think they call them something else at Bristol. What is it? TSR, teaching support. Anyway, Annie Boja and Andrew Shannon. I don't know if either of you in the audience today. No, okay, we'll meet them on Friday then, or whenever the lab is. So you all know the format for this course, right? There's going to be 7 weeks of lectures, and then there's going to be some weeks where you'll complete a coursework, and at the very end you'll do a final. And so the coursework is half of the final mark, and the exam is half of the final mark. This is my first time doing the whole lecture series for the full term. I'm kind of a new lecturer, so I might need some of your help in sort of like interactivity, keeping me on pace in the lectures. Just interrupt me if something's going really weird. Last year we had like a good crew, because I joined kind of like halfway through term. We had a good group of people that would sort of go back and forth and sort of keep things exciting for questions and stuff. Oh goodness. Yeah, I know, yeah. Right, so if you're wondering where to find something, it should be on this GitHub link. Let's see if this works. It works, okay. Yeah, so I put everything up on this GitHub, which is on the SEMT group on the GitHub website. And everything's up here. So right now, even the slides are up here. So the slides I'm showing today, everything's going on here, needs to be on here. So if there's something that should be available to you that you can't find, let me know and I will fix it. But everything should be on there. As far as the textbooks for this course, so the curriculum is going to be largely based on a design of the course by Connor Houghton, whom many of you may know, but he's a professor in our department. And most of the lectures are also going to be accompanied by a reading from his course notes. And the general idea here is that the slides are mostly the definitive sort of, is this on the exam? Well, is it in the slides? Then yes, it might be on the exams. But the readings are meant to parallel that. And these readings were sort of controversial in the feedback, and sort of some students loved these readings and some students did not love these readings. And so take a look at those. Let me know whether you like them or not. But the course notes are designed to accompany the course. And all right, sorry. Oh. As far as textbooks, so the two core textbooks which you might want to download are the Neuronal Dynamics by Gershner and colleagues and Theoretical Neuroscience by Abbott and Diane. So the Gershner book is pitched at a little bit higher level, but it is also a bit newer. Do we have it? whereas the Diane and Abbott, I think, is still the, what would you call it, the canonical sort of computational neuroscience textbook. And these are also all on the GitHub with links and everything that you might need to find the chapters. Yeah. Testing. Right. Well, we already covered this. Yeah. All right, so I think I'm about to launch into sort of a monologue with slides. So if anybody has any questions actually about like administrative things or organization or scheduling, now might be a good time to ask. I feel like the course organization is pretty straightforward. Coursework, exam, everything's on the GitHub, including these slides, which contain the emails and so on of the course staff and stuff. You need to look that up. Oh. I have a question. What made you sign up for this course? Did you know you were getting a completely, I guess, green unit director who's going to be really strange with the pacing of the lectures? Yes?
My mate got a first.
What?
My mate got a first.
Your mate got a first?
Yeah.
How did your mate get a first?
Well, he got 100.
Oh, yeah, that one. Yeah. The marking was blinded, so formally I do not know who your mate was, but I remember that paper. I'm like, okay, so I'm going to try to make it a little bit harder this year. So the first is going to that first is going to be a 95. I think that's what's going to happen. We'll see. There'll be review sessions back and forth. You know, just ask me. We'll get through it. Okay, so I wanted to start. Okay, so. Why brains? Apparently I thought this was a clever slide title. Let's see. Does anybody know why we have brains, generally speaking? Survival. Yeah, that's probably about it. I'm not sure that the why is that important. If you just want to know like how they work, you can study computational neuroscience without worrying too much about why brains evolved. But yeah, so you've got this 1 to 2 kilogram thing inside your head. It's using a lot of your energy, so it must be worthwhile, you know, evolutionarily speaking. Although I'm not sure how much longer that's going to be true with the way that the news has been going for our species, but yes. So we've got, is 10 to the 9 millions or billions? Goodness. Thousands. Millions, yeah, billions of neurons in our brain. Mice have millions of neurons. Our brains have billions of neurons. What are they here for? I quite personally like, I mean, survival, right? Yeah, but for me, one of the more like clever takes on you have brains to survive is based on, you know, Claude Bernard's work, which was at the end of the 19th century, when he introduced these ideas in physiology, which would later be called homeostasis. And it's about regulation of sort of the internal states of the body. This is for the survival of the body. Yeah. Oh, goodness. So I can see that my chaotic lecturing style, or rather grossly underprepared lecturing style, has promoted several conversations around the room. And I have a question. So for me, I personally didn't encounter Claude Bernard's work until quite recently in my life, like a couple years ago. And for some reason, I found this incredibly profound, incredibly insightful. And in particular, this idea that like as you elaborate on what the brain does and make it more and more complicated, you aren't necessarily like getting more detached or more dissociated from the world. What you're doing is you're gaining more and more ability to model the statistics of the world. So it's sort of as if, yeah, but I don't have anything else. Yeah, next time. Does anybody familiar with this origin, Claude Bernard's work? No? Okay. Well, it's good to know. He at the time he called it this idea, the milieu, did anyone speak French? I don't pronounce it right. Milieu interior, the interior environment. And this is really sort of where some of these ideas that would later become computational neuroscience come from. So I guess basically pre-19th century, we're starting to get mathematical physiology. People are starting to become more quantitative with how they understand how the body works. People are looking at sort of how the heart pumps blood and how you can model that with force and pressure and flow. And again, towards the end of the 19th century, Claude Bernard comes along and says, well, basically everything's about regulation. You've got something you want to stabilize. You're going to basically do negative feedback to keep that stable. Of course, the term homeostasis will be coined a bit later. And I would say that the sort of field itself emerges in the middle to early 20th century when we start to be able to do physiology and neurophysiology in more quantitative ways. So people start to record the electrical signals in the brain in the early 20th century. And then around the middle of the century, and we're going to study this model in this course, Hodgkin and Huxley model of how neurons make spikes and communicate, really take off. And this is one of the most successful results of all of computational neuroscience. And then, also around this time, you get this cybernetics movement, which is where a lot of the more abstract representations of neural computation, the idea that you can have maybe something that's not quite a neuron, but sort of like a neural network inside a computer, and you can train those to do things, and maybe it is thinking a little bit like the brain thinks, and so on. And so this starts in the middle of the 20th century, but continues right to this day, including this immense takeoff of AI over the past 3 years, sort of ultimately rooted in these sort of connectionist ideas. So that is as far as I'm going to cover for the historical context of this course. This is a great quote from Neil Simon. So Neil Simon is or rather Alan Neil, Simon's a different guy, but this is, again, mid-century, more of a computer scientist, so more of working on, developing programs and sort of the AI that runs on von Neumann architectures, old symbolic AI and stuff. But anyway, they were quite optimistic that they were going to solve the brain or sort of compete with human intelligence within a few years in 1958. And for a while, people thought, this is going to be great. And then they tried to build these machines. It didn't work. It was very hard. And we have all these AI winters. But arguably, I would say that by now in 2025, this is basically correct. I mean, who here has not talked to a large language model? Everybody's using AI, right? And it's pretty good, right? No? I have actually run most of the exam questions in coursework through the AI. And they do make sort of weird conceptual mistakes, but in terms of getting like everyday math and basic pieces of reasoning, it's all there. Oh, okay. So, let's see. Looking at this picture. What levels are we going to model at? So, I would say, I'm missing a slide. You know what? I want to just go to a blank page. I think I want to draw something. Let's see. So we've got biology. Let's see. So computational neuroscience, at least the way I do it, is mostly about trying to understand biology. So I want to understand how the brain works, how all these little pieces, all these things that start with single molecules, you know, ions and dissolved in water and so on, how the interaction between all of these elements gives rise to everything that the brain does, so our complex, rich, conscious waking experience. And that is actually a very difficult thing to understand clearly. So why computational neuroscience? Well, a lot of it is about trying to build these mathematical metaphors or mathematical models, which are, in a sense, grossly simplified, throwing away most of the important details, but not simplified so much that they become completely wrong. And the goal here is not so much that I'm going to build a perfectly realistic biophysical model of the brain, you know, that I can use to simulate it to every detail, but to make precise a mathematical story about what a certain system is doing. And at the same time, of course, heavily involved in this is running computer simulations to sort of explore the mathematics with computational analysis of data to try to figure out what the brain is actually doing. And the main sort of crux of this course is you're going to be repeatedly presented with problems that are going to ask you to move between these spaces. So there's going to be some sort of biological system, there's going to be some sort of reduced mathematical model, and you're going to be asked to simulate that mathematical model and then comment on how the results of that simulation do or do not relate to the underlying biological system. So that is the sort of ritual that we will be doing in this course. And this is, yeah. So this slide is here from the Gershner book, and this is just pointing out that the field itself covers multiple scales, and in this course we will also be covering multiple scales. So we have a little bit of this, low-level biophysical stuff. We'll start with that. Yeah, some dendrites, barely. There's definitely going to be some spiking or cellular levels stuff. Many of the, there's going to be a lot about like Hopfield networks and perceptrons and stuff, and these things are all sort of very abstract, where you're just sort of thinking about neurons that can be on and off. And none of this stuff. They might get to field models at the end if we're lucky, but yeah. So the stuff on the right is more cognitive science, and that's more Sean's course if you signed up for that one. And I reckon I put this slide here originally because this was supposed to be that sort of triangle diagram I just drew, saying you're trying to understand the biological system, so you'll build a sort of cartoon mathematical story, and then you simulate it, and you sort of go back and forth between this and say, can I distill the important features in this simulation to make my theories about how this thing works precise? So there's really more, it's not really possible for us to cover everything that you might need for computational neuroscience in this course. So I'm trying to figure out what do we actually have. So you won't be simulating anything at the level of single molecules. People do that though if they're simulating like protein turnover and protein dynamics in a synapse, because some of these synapses in your brain, they have on the order of like tens of receptors. And so at this point, you might run a computational simulation that really is just storing a number between like 0 and 20 for the number of receptors that are working right now. But we will not start at that level. I'm going to make you do a little bit of mass action kinetics models for neurotransmitter diffusion and stuff like that. So this is chemical reaction modeling. This is going to be a huge part of the course, these conductance models. Leaky integrated fire models, we'll get to that soon, either today or tomorrow as things go. Rate neurons, at the end of the course. Poisson neurons, this is going to come in more in the coursework. These are mainly used, these are more often used for data analysis. And binary neurons, again, in the middle of the course. So, let's get to it. So this week, we're going to run the introduction and just try to get through basic neuroscience. So there is this slight issue, which is that you will need to memorize some random facts about biology, which are not directly related to anything computational. Just, make some flashcards for that. I mean, it's important that you know generally the names of the things in the brain. So that's what we're going to partly cover this week. We're going to try to get into the electronic computation of neurons as quickly as possible. And then by week 8, we're going to switch over to doing the coursework. All right. So again, gosh, I'm sorry for the really, really poor charisma level in the lecturing right now. I'm just not feeling the flow. Anybody want to rescue me? No? No one's going to? Yeah, it's one that's going to be one of you up here. You'll see how it feels. Oh, man. It's good, though. We're only 20 minutes in. I mean, what am I supposed to say? I mean, I'm down here with the microphone monologuing. I mean, when they give you this training for lecturing, they're saying they're supposed to make you do activities, make you go into groups, communicate with each other, I'm like really bad at that. So if you guys want to do a day where we actually have like think, pair, share questions and stuff like that, I'm happy, but I'm going to need your help organizing that and making that go well. But we need to get into the course content, because actually the first 2 lectures do contain stuff which will be on either the coursework or your final. So yeah, for legal reasons, we need to continue. So it's the first sort of content slide deck starts with the question, brains, what are they? And this is just brain facts. How many of you know facts about the brain, what the brain is made of, basic stuff? Shaking, really? No. Yeah? Are these words, have you seen these words before? Because I feel like it's hard to get to be an adult in the world these days without hearing words like gray matter, white matter, cortex, so on and so on and so on. And the other reason you might want to know these anatomical terms, if you have to think about your brain as like some sort of gigantic, I don't want to say data center, but some sort of like weird, complicated system architecture, right? So if you're an engineer working on that, you'd want to know like, oh, that server handles this, and that server handles that, and that fiber optic line is doing this, right? And if I'm hiring you to maintain it, I want you to be able to use the names for like the correct fiber optic line and so on and so on. So we're going to just cover some basic vocabulary for that. And it's just like learning like, okay, your computer has like a CPU and it's got some sort of, RAM and stuff like that, right? So the first thing to check is you basically have neurons that are the things doing the computation. And then you have cables that connect these neurons together. And yes, okay, to be fair, some computation happens with those cables, in those cables, but they're mainly about communication. So the actual cabling is this white matter. These are these, and it's white because actually in order to prevent the electrical signals from sort of spilling over or leaking out of the cell, the neural processes are wrapped in fat. And that's this myelin insulation. So maybe I should write fat. I mean, that's a bit of a crude way to put it. What I should say maybe a lipid-rich, multi-layer, membrane, or something more technical. But yes, okay, so fine. We've got, and the gray matter is of course cells. So if I were to zoom in on this sort of mouse, I would say, okay, I've got some actual neurons in here, making up here, and then they're sending their cables down into here. Okay, so that's loosely what the brain is made of. It's got the cell bodies in the gray matter, and then they're sending their cables to this white matter. This outer layer where the cells are, this is the cortex. And because I'm sort of labeling diagrams and so on here in a lecture, you might be asked to label diagrams or something on the brain facts section of the final, stuff like that. So, come back to these slides later when you study. In large animals, right, the brain's kind of folded up, and this is just so you can get more actual cell bodies in there. So what they'll do is, instead of having a smooth brain, where the cells are all here and they're sending their cables through here, so really small animals have brains like this, like mice and shrews and stuff. But of course, in our brain, just to keep more going on, the extra gray matter is sort of crammed in there, and to do that, it's folded. So a gyrus is like the part that goes up. Gosh, there's got to be a better way to put this. sulcus. Does anybody take like Latin or a Romance language, or is it Greek? These roots?
Greek. Yes, it's Greek.
Yeah, it's Greek. So what do these mean in the original?
It means turns.
Turns, okay. Turns. Cool. And sulcus? Is that still in use in modern Greek, or is it too old?
That might be Latin actually.
I mean, it should be valley, but I can't think of any other loanwords in English that have sulcus, like those sounds hiding inside of them, so I can't guess. Well, if anyone looks this up on like Wiktionary on their phone, let us know in this lecture. Most of the anatomy terms are just describing what you see, but in Latin or Greek. I think I even added some notes about this in the Connor's course notes. Like, there's a place in the brain called the substantia innominata. Does anybody want to guess what that basically means in English? Like unnamed stuff. Like, it's just like the anatomist got to the end of the day and it's like, you know what? Fine, I'm going home. Oh, have you heard that? Lobes of the brain. Yeah, I'm trying to think how to explain this, right? Because on one hand, you want to say that the brain is resilient, the brain is redundant. If you, it can sometimes adapt to tissue loss and so on. On the other hand, it really, really can't, especially as an adult. And functionality gets specialized to various sections of the brain. So if, you know what, I forgot to bring my brain model. I'll bring that next on Thursday, brain model. So next week we'll have a brain we can pass around and take apart and remove the lobes of the brain. But the general idea is that, yes, you know, if I give you damage to temporal cortex, you may lose your language skills, or you may lose the ability to recognize or name objects. If you get damage to occipital cortex, you will have what's called cortical blindness. And there's this idea that you get functional specialization in different lobes of the brain. And what determines this functional specialization is, in part at least, based on what that lobe of the brain is connected to directly during development. So occipital is the locus of vision, mainly because the signals from the eye are, among other places, going there first, in a certain sense, into cortex, and so this takes on the responsibility of interpreting the visual world. Of course, if you're not using your eyes, you can use occipital cortex for other things, but this connection, if you develop with sight normally, is what assigns sort of visual processing to occipital cortex. So I'm trying to figure, I do not know how to teach this in an engaging way yet, because this stuff is going to potentially be on the exam. So you all could just make a note card and sort of say, okay, what does parietal cortex do? It's some sort of multisensory integration. You can see it's sort of at this crossroads where the other lobes of the brain are all sort of adjacent to it, and so on. So, Has anyone heard of a guy named Phineas Gage? Steel Rod guy. Tell us the Steel Rod. Yes, tell us the story. Do you know the story? I can tell the story, but
I do know the story. I mean, I know the part where he sells his brain to a bunch of universities, and then none of them actually have any claim, and he used all the money.
That is brilliant. I didn't even know that.
That's why he got a steel rod through his brain, and it actually didn't severely impact.
Yes, apparently you don't really need the brains. Yeah, it's just kind of there.
His brain just like folded around it, and he could still do everything okay. So yeah, he sold his brain after that to loads of universities. He promised them.
I never heard that part of the story. Yeah, so the idea, Phineas Gage, I think he was a railway worker or something, right? And I don't know whether they were blasting a tunnel or doing something, but there was some sort of use of explosives, and it didn't go quite right. And he managed to get like a rod like into his orbit and out through his brain of steel, just explosively. So that thing went through his brain. I don't know exactly which part of frontal cortex it knocked out, but there was tissue loss. It wasn't just like, oh, your brain gracefully folded around the bar. It's like, no, I just punched out part of your frontal lobe. And, he was fine-ish, but the general story is that he became really unreliable and just not like very sociable. And so it turns out that a lot of your executive function, higher order social abilities, conscientiousness, social rules, emotional regulation, that's all handled in the frontal lobe. And our respective frontal lobes probably achieved that to varying degrees of success, but Phineas Gage was kind of surprisingly okay. We are also going to talk about a patient, H.M. Henry Mawson? Gosh, I can't, Mawson? Or whatever. Trying to remember when that is. That's going to come up in the hippocampus section, but all right, guys, check out this brain. I need a different color thing. So someone knew the story of Phineas Gage. Does anybody know the story of Tan or Broca's area? Is this new? It's not as colorful. Apparently scamming a bunch of universities regarding a brain donation gives you a much more memorable brain damage. Did anybody see that TV show Game of Thrones? Anybody back then? Yeah, there was this one character who could only say Hodor, right? Due to magical time travel reasons. But in real life, that happens when the part of your brain that is responsible for sequencing the delicate actions which move your lips and mouth and vocal cords to produce different words. If that part of the brain dies, then you will struggle to speak with much fluency or complexity. Paul Broca was a neuroscientist who encountered a patient who could only say the word Tan. Upon Tan's death, they dissected the brain and they found out there was a hole in it. And it was in a different part of frontal cortex, which is now called Broca's area, and this will be on the exam. So the idea here is that actually the idea is pretty simple. You need that part of the brain to speak. So with Phineas Gage, we talked about the frontal cortex being about regulation, planning, and so on. But as you sort of get more posterior, more towards the back, you get into more and more motor-like functions. So you can almost think of these cognitive rules as sort of the most abstract form of movement or movement planning. And so maybe you start at the very end with sort of like desires, emotional regulation, and goals. You get to sort of like intermediate plans, like I'm thirsty, I should probably get a drink of water. And then that is like, okay, well, I guess I should, plan to use my left arm to pick it up in the premotor system. And then when you get to primary motor cortex, this is the part that actually sends the output. So the primary motor cortex for my left hand sorts it out. And so basically, the Broca's area is a little bit like the premotor region for the lips and the tongue in relation to speech. It cooperates with another area, which is also quite important, which is, for some reason, not on this slide, although it should be, which is Wernicke's area. Can anybody want to guess what happens if you lose Wernicke's area? I just want to check. Did I actually circle Wernicke's area? This does cause me some concern. Goodness, I feel like it's got to be like around here, actually. No one wants to guess. It's okay. So these two areas are sort of cooperating with each other, Broca's and Wernicke, when you speak. And so Broca's handling the motor sequencing. So if you lose that, you can't really speak, but you can understand speech just fine. Conversely, if you lose Wernicke's area, you can speak fluently, but the words you're going to speak don't make any sense. So they're still words, It's just a complete random substitution of like a related word for what you meant to say. And that's because the part of the brain that is supposed to tell Broca's area, like how to inject useful meaning or useful communication onto that stream of words, is not there. So this is called fluent aphasia because of course you can speak fluently. Aphasia is again that's probably Greek as well, right? Is for like some sort of issue with speech, so you can't speak. Well, you can speak, but it's not. But you can produce sounds fluently, but they don't really mean much. So, we've got some other brain areas we're going to encounter. Let's see. All right. So we are actually going to look at models of the hippocampus. That is a big part of the course. We are not going to look at the amygdala, apart from today. You might want to remember that it has something to do with fear conditioning. I might add a fear conditioning learning rule at the end if we have time. We'll see. Nice fact about the amygdala. If your amygdala dies, you do not experience fear normally. So if you were to have a disease where your amygdala calcifies over, you would not, actually you would probably be a much better lecturer, I would imagine. But sadly, I think we can't all be so lucky. So anyway. Cerebellum as well. We're going to talk about that a bit later on, around the same week we talk about the hippocampus. These are relevant because the hippocampus and the cerebellum are sort of areas where there actually are decent, but rather abstract and simple models of sort of what the neurons there do. And so they're a good thing to bring up in a undergraduate computational neuroscience course because the models are accessible and they're arguably not wrong, even though they're quite simple. People do tend to forget about the insula. It's down there inside of our brain, so usually, these main lobes of the brain, occipital, parietal, frontal, temporal. But if I were to cut into the brain and pull off that chunk of temporal, frontal, and parietal cortex, I would find that there's a whole other lobe down there. And this does a lot of stuff with sort of emotion, taste, smell. There's also some stuff about language and motor planning and stuff going on down there. There's just a lot of gray matter packed in there. We've got a whole other little bit of the brain underneath the rest of the brain there. So the hippocampus and cerebellum I've moved to later when we actually talk about the models related to that. But you should also know that the brain is not just cortex. But that there is a lot of machinery in the middle, and we actually do not understand this machinery that well. So this is a slice of a human brain. It's been stained, and I don't know exactly which stain they use, but this stain is actually coloring the white matter black, just to create contrast. So everywhere you see is lighter here is a place where there's a bunch of cell bodies, and so we can see cortex. This is cerebellum down here. It also has, cortex and white matter. But what else we got going on in here? So we can see some of the basal ganglia, the caudate, putamen, globus pallidus. I do not think I'm going to ask you to specifically remember these names because there's not enough time to really go into depth about what each of these little different basal ganglia do. And the problem is also we don't actually know what they do, so this is the issue. Very well. So one thing that will be on the exam, though, is there is one especially well-studied part of the basal ganglia for an incredibly common disease, and that is the substantia nigra. And anybody want to guess what that means? You may already know. Latin for, it's like black stuff. Again, dark stuff. It's because the neurons there, when you're dissecting, just appear a little bit darker than the surrounding tissue. And I think there's actually a reason for that. I think it has something to do with their metabolism for producing dopamine. And these neurons, their job is to produce dopamine. So they have these axons that go back out into the brain, out into motor cortex, frontal cortex, parts of the basal ganglia, and deliver a dopamine impulse. And if those cells die, well, you get Parkinson's disease, which is not great, to be fair. So the main thing is that it impairs movement, so you get stiff. You might get some tremor, and it becomes hard to initiate movement. But this dopamine and these systems also have a cognitive role. So for most of these motor systems, you can find, motor symptoms rather, you can find some sort of cognitive analogy, like maybe not being able to switch planes as quickly or to think as fluidly if you're not able to move as quickly or fluidly. And again, this is potentially on the exam, so we'll see. Personally, I am quite frustrated that we do not understand exactly how the basic ganglia work. There is a slight problem with teaching computational neuroscience, which is to understand any topic well, you're supposed to ask, what do they say, 5 whys? Like, well, why does he have Parkinson's disease? Well, because neurons in substantia nigra died. Well, why did that happen? Oh, metabolic stress. Okay, but why did that cause that symptom? But once you get down to about the 3rd why in neuroscience, you're just into open research questions. So some of these things just do not have clear answers yet. Maybe some of you will solve them. We will see. So this concludes the brain facts section of the first lecture. And now we have to move on. Does anybody want to talk about brain facts before I switch topic for the day? Okay, brain facts. They're on the, yes.
I just have a question. Has anyone ever tried to remove the amygdala?
Probably. Anybody want to Google that real quick? What? I mean, I'm sure we've done it in rats, right? So I'm going to go ahead and say that intentionally removing the amygdala in a human would not generally be considered ethical or appropriate. And there are a handful of stories which have made it into the textbooks where people have done things to humans, and they usually come with a paragraph saying, that was actually a really bad thing to do in retrospect, and we're not going to do that again. Like frontal lobotomies, or removing all the temporal lobe to cause permanent amnesia. It's like, okay, not the best. So I have never heard of a story of someone intentionally removing just the amygdala, but I don't remember her name, but there is, in the textbooks, it's usually this one woman who had this, just spontaneously had her amygdalas calcify and die. And so she was well studied. So I think that there's never been a good reason to do that. It wouldn't be ethical, but incidentally speaking, there have been patients where there's a disorder or disease that just killed off the amygdala, and that's where we get this thing of like, they just don't really have a fear response like we would understand it. I mean, I'm trying to think if like the amygdala does other things, because it holds fear memory, instinctual memories. I've never really thought about whether there's other types of instinctual memory that aren't quite related to the emotion of fear that might require the amygdala. If anybody reads up on that, let me know, or remind me to Google that. It's not part of the course, but I would also like to know more about this. Okay, right, all right, so the brain, do we all know already that the brain is made of neurons? Is that, okay, that's good. You've had some sort of biology textbook, right? There was a period in history where we weren't quite sure that the brain was made of neurons. And this goes back to Golgi and Ramon y Cajal. I can't remember exactly which century they worked in, but when people started cutting up the brain and being able to visualize what's going on with these cells, they started discovering these massive labyrinths of little wires. And of course, these are the wires that carry the electrical signals that the brain is using to compute on. And these wires are growing out of neurons, but for a minute there, we weren't exactly sure whether these wires connected up between cells. And I think Golgi was a fan of that, thinking that there was what they called a syncytia, where, sort of the tubes from one neuron directly grow into the tubes of others. And that ended up not being the case. For the most part, Neurons communicate through synapses, where they're technically separate, but they have this electrochemical connection. Of course, later on we would discover that there actually are synapses that do directly connect neurons, so the syncytia idea was a little bit correct in the beginning. But just look at how nice these are. Apparently these Ramon y Cajal drawings, there was a period where people were just stealing them from the library where they were stored and taking them home because they were so beautiful. And so all the ones that survived got stamped with this library stamp to sort of prevent you, so that if you steal this, we will know that you took it from this library, because they had to stop the theft of these things. So we need to know the basic components of a neuron to model a neuron. How many of you have actually already covered all of this stuff? Okay, we got 2, 3, 5% of the class. Okay, so it's easy. I would say also in biology there are some weird neurons out there. So as much as I'm trying to say that this is what a neuron looks like, feel free to go away and find out the bizarre configurations you can get. Not all neurons are organized into clearly defined soma or dendrites, as I would do in this picture, or rather axons and dendrites and so on. But yeah, no, the general picture is right. You've got a cell, so a cell, it's a living cell. It's got a nucleus. Do I have a pen? It's got the nucleus with the DNA that's making the proteins. And of course, these have to get shipped out along these wires to the connections that the neuron is making with other cells. So that's what's going on in the soma. The other thing that's really important for the soma is that this neuron is sort of like an electrical component, right? So there's all sorts of inputs coming in. Here, on this big apical dendrite, there's inputs coming in on the basal dendrite, and these electrical signals are sort of being transformed through an electronic circuit, and they all end up changing a voltage at the cell body, and in a way the neuron will decide whether or not to emit a spike, which we'll get to in a minute, what spikes are, based on that. So that's the other thing to notice about the soma, is it's sort of the convergence within each neuron of these computational signals. For the most part, the neurons we're going to think about in this course all have dendrites. And I just talked about dendrites, apical dendrites, basal dendrites, and so on. There's other types of dendrites depending on how your cell is shaped. Lots of different morphologies and shapes that cells can take on. The Purkinje cell so the neuron on the right here is a Purkinje neuron, and it's got one of the most beautiful and charismatic dendritic trees of all the neurons. And we will talk about that more when we study the cerebellum. This guy is like, well, I don't want to get ahead of myself. More later. Right. And then axons are the wires that send information away from the cells, generally speaking. So we've got inputs coming in on the dendrites, integration of those in this electronic network that is the dendritic tree, converging at the soma, where the neuron will make a sort of decision to spike or not, which is to say, will it send an electrical impulse downward to communicate with other cells, and that impulse travels along the axon, and at the end of the axon or along the axon are these synapses, and of course these synapses in turn connect to other dendrites. And that is basically, yes, that's basically what's going on in the brain. This can get rather complicated. So these cables can go long distances. They can be, millimeters or meters. Dendritic trees can be pretty diverse, depending on what the cell is doing. And this is partly because the sort of geometry of this determines what inputs that neuron can connect to. So some of the connectomic or circuit diagrams may inform where you send the dendrites to create these different patterns, but also the different geometries of different electronic properties. So on one hand, it's tempting to think of a neuron as sort of like the universal transistor of the brain, and that is sort of true. But if you look at this picture, you can see that there's this immense diversity in the design, and the specific sort of analog computations that each cell does can be quite different. And it's not just the morphologies that differ. Like, different neurons will express different ion channels that respond in different ways with different time constants, which will change how they transform inputs into outputs. So, and we are not done discovering the diversity here. So we have not fully characterized all the bits of the brain. There's lots of missing knowledge. By and large, I wanted to tell you that axons grow out of the cell body, but as I was preparing this slide deck, I found in my Google search results a paper which says sometimes the axons just grow straight out of the dendrite. I mean, it's close to the cell body, but so it's very difficult. I wanted to sort of say, okay, there's an axon, so I'm a dendrite. All neurons sort of have these, except when they don't. But for this course, all neurons will have these dendrites that take in inputs, axons which transmit outputs, and we won't have to worry too much about it. Here are some synapses. Let's see. Okay, so there is one detail which we will need to cover, and I'm trying to get this out of the way in the first week because it's often considered one of the less interesting bits of modeling to have to deal with in computational neuroscience, which is dealing with the electrical potential, which is allowing neurons to sort of have a voltage difference across their membrane and to use that voltage difference to drive electronic computation. And the basic idea of what's happening here is that our bodies and our cells contain lots of different salts, sodium chloride, potassium chloride, magnesium, calcium ions, and all that stuff. And these carry electronic charges. So something like chloride might have an extra electron. Sodium ion is missing electron, so it's positively charged. And so what a neuron does is it has these transporters in the cell which consume energy to pump charged ions across the membrane. And what this does is it sets up a situation where the concentration of particular ions is slightly different on one side of the membrane or the other. And that is essentially a chemical battery. So, I mean, it's really not much different from like a AA battery that you would use to convert chemical to electrical energy in the Vegas sense. Yeah, so what a neuron is going to do after it establishes these various ionic concentrations, and each ionic can sort of act like a different battery, is it'll then open up pores that allow these chemicals to go across the membrane, currents to flow, and that flowing current will drive the neuron's voltage in various directions. Okay, so I'm sensing we should wrap up just so the next lecture can get in, but I wanted to leave you with the only effective mnemonic I've ever seen for remembering which ions go where, which is invented by Connor Haughton. So I found this immensely difficult to memorize as a student, but thankfully you have this beautiful picture, which is a bit weird, but it'll help you remember that sodium and chloride are outside because they're in the sea. And for some reason, and potassium is in the inside because it's in the UK. Yeah, I don't know. This is supposed to be the neuron. It's in the slide deck. You'll come back to it later when you download this to study. I look forward to seeing you all on Thursday. Thank you so much for helping me get through this.